Hello, everyone. I'm Hisashi Yamakawa from Kazusa DNA Research Institute. Today, I will show you our environmental DNA analysis system in ANEMONE. This is a picture of our institute. Yesterday, some of you invited us and have a tour of the facility of KDRI. I hope you enjoy it. あ、そうか。こうですね。Oh, I'm sorry, but I will skip some slides because in the morning session, Dr. Michio Kondo already showed the background of our analysis. Okay, I will start from this slide. The eDNA Society in Japan has published environmental DNA sampling and experiment manual that explains the procedures from environmental DNA collection to analysis. Environmental DNA surveys in Japan are often conducted in accordance with this manual. ANEMONE also follows this manual, essentially. We will explain the procedure for environmental DNA analysis at ANEMONE. The basic workflow is the same as that of commonly performed environmental DNA analysis. For sample collection, a water sampling kit containing the necessary consumables will be distributed and on-site filtration will be performed using cartridge filters. A distinctive feature of this method is that, as a rule, samples are collected by filtering using 2 filters at each location. This not only increases the amount of filtration, but also allows the other to act as a spare if one filter is lost due to some kind of problem. Purification is performed using the popular Kaizen DNA Easy kit with elution in 200 microliters. The 2 samples are separately purified and stored and then mixed using PCR to use as a template. The PCR amplification target for fish is the MyFish region. PCR is repeated 8 times. It may not be necessary to repeat the PCR so many times, but as we use 8 tube strips with lids for the work, we have limited it to 8 repetitions for ease of use. This process increases reagent costs and analysis time, so we would like to reconsider it at some point. Samples after PCR are promptly frozen and stored. The 8 PCR samples are mixed before purification and then purified on a single column. After purification, amplification of the target band is confirmed by electrophoresis and quantified at this point. Based on the quantitative value, the amount of template to be input into index PCR is determined and an independent index is assigned. Here, a dual index system is used. After the reaction, 1.5 of 96 well plate is poured, concentrated on the column, and The target band is excised and purified by electrophoresis. The excised and purified sample is concentrated using a column, and the concentration is adjusted before running the NGS run. For NGS, we use the Illumina MiSeq or NextSeq 500. These are random flow cell machines with a relatively low index hopping rate, which we will briefly discuss later. We also own an even larger machine, for example, the NextSeq 2000. But since it is a pattern flow cell machine with a high hopping rate, we do not use it for metabarcoding analysis. From here, we will add more information about the characteristic of anemone's analysis methods. As mentioned earlier, when collecting samples, we basically use two filters, each processed 1 liter of water. Naturally, we also need to be mindful of various types of contamination and take care of the contaminate and protect sample. There are many different methods for sampling and analysis, but anemone used a marked method. Cartridge filters are used to collect samples. The DNEZ kit is used for extraction and purification. The target of analysis is the MyFish equivalent region, mitochondrial 12S or 16S, depending on the species, but we expected to use other regions depending on the species in the future. The NGS used is an Illumina short-read sequencer. An important point when analyzing environmental DNA is that the DNA is extremely dilute and that a large number of different samples must be handled simultaneously. Therefore, the most important issue is to take measures to prevent DNA contamination from the outside world and cross-contamination between samples. During DNA extraction, we place particular emphasis on measures to prevent external DNA contamination. So the environmental DNA extraction room is built to clean room specifications and is basically isolated from the outside world. When entering the room, staff must wear special work wear and disposable gloves. Before and after work, staff also thoroughly clean the lab benches, cabinets, and the equipment used. The DNA extraction space and the first PCR 後ろにあるですね。First PCR construction space are also isolated to prevent contamination as much as possible. As we have no control over the air conditioning, we cover the exits to allow the wind to escape. The filters are dirty materials brought in from outside, while the extracted DNA is thin and clean. In order to handle these two simultaneously, the workspace is separated. The filters are handled inside a safety cabinet, and the extracted DNA solution is handled in a clean environment outside the cabinet. All workers wear clean suits, just like when working in a clean room. For column work in DNA extraction, we use equipment that creates a local clean environment, and work is carried out within the effective range of that equipment. The table coach creates a gentle breeze with clean air, eliminating dust that falls from above, creating a clean space. When the space is monitored with a particular counter, while it is operating and while it is not, it's clear that when it is operating, there are absolutely no fine particles floating in the air. When assembling the PCR reaction, we take the utmost care to prevent external contamination. We use a clean bench to assemble the reactions. The reaction mixture is mixed inside the bench and then capped before being taken outside. PCR enzymes are generally handled on ice, but bringing ice in can cause contamination. So we have recently installed a device that can cool the reaction without ice. Furthermore, as the amount of liquid handled from this point onwards is very small, we have installed and used static eliminators in all work areas to prevent the solutions from splashing due to static electricity. It is important to avoid freezing and thawing dilute samples as much as possible, as the composition and the degradation caused by freezing and thawing can affect the overall concentration. We store these samples in a freezer that does not automatically defrost. After the first PCR is complete, work will be carried out in the same level of work area as for regular NGS sample preparation. The points to note regarding work are basically the same as for NGS sample preparation. No special measures will be taken here. NGS analysis involves profiling using amplicon sequencing, and currently analysis is performing using an Illumina short-read machine. Kazusa has a variety of NGS systems, but currently only uses the MiSeq or NextSeq 500 due to the index hopping that is unique to Illumina machines. The decision has already been made to continue the MiSeq system, and we are planning to switch to a new one while the current system is still in good condition. ちょっと早すぎたかな。だいぶ時間が余りそうだ。よいしょ。Index hopping is a phenomenon in which index sequences used to distinguish samples are recombined with other sequences and is unavoidable due to the nature of Illumina sequencers. As a last resort, unique dual indexes were devised, but this reduces the amount of data, and when analyzing a large number of different samples simultaneously, as in ANEMONE, there may not be enough indexing to begin with. Therefore, we are currently processing with analysis using the combinatorial dual index method prepared for some index hopping. あれ Oh, that's all for the analysis of anemone samples. As for the future, we have already introduced several new NGS machines from companies other than Illumina. and we will transition into these. We are also in the process of introducing robots in order to increase our immediate output by about fivefold. In addition, we are developing new protocols to resolve various issues inherent in our current standard protocols. If we make any changes to our current procedures, we must always consider what will and will not change compared to the past. Otherwise, it will be difficult to compare the data we have accumulated with new data, and this would defeat the purpose of ANEMONE. Thank you for your attention.
Thank you very much, Professor Yamakawa. So we'll be happy to take a few questions. All right, please.
Thank you. Thank you for the talk. It was really interesting. You mentioned about PCR replicates, and I was quite impressed at the amount of replicates you have in each PCR step. So each replicate, you need to add 1 or 2 microliters of DNA. You might be diluting it. But when you think about applying additional assays in the future, would you have enough DNA to do that if you have the amount of replicates? Or would you consider about maybe reducing the replicates or diluting even further so you have more volume? Have you thought about that kind of balance?
Now we use 2 microliters per reaction. So 8 replicate needs 16 microliters. It is very large. And so now we have not まあ対策はしてないんですけれども、but our new protocol may not use that so much. So in the future, 多分そんなに作らないということになると思いますがというところです。はい。英語があまり得意じゃないので申し訳ありません。
Thank you.
Thank you.
また詳しいことはまた後ほど。
Next question.
I just heard you have many minutes for a Q&A session. So just I'm asking. You have utilized so many sequencing systems across the companies, right? And do you have any comparison data using recent ABITI and DNA-seq T1? And also actually you conventional MiSeq system for eDNA research at this moment?
No, I don't.
同じサンプルで比較したデータってまだも取ってないっていう。
これから取ります。
Thank you so much.
Thank you. Another question? Yes, please.
Thank you so much for explaining the protocol to us. I have 2 questions actually. The first one is that you controlled for contamination very carefully. So do you still have contamination after controlling for all this? That's the first question. My second question is about your third point on reviewing the protocol. So in my lab also, we keep improving the protocol. And like you said, sometimes the data is not comparable because you're not using the same protocol. So maybe can you share with us what you consider when you change the protocol? Thank you.
For the first question, yes, sometimes, very rare and sometimes from human DNA, maybe from workers. Previously, we detect mouse DNA from these samples, but the workspace is near the mouse なんだ飼育室飼ってるところで近かったんです。なので、so the infect その影響かなと思うんですけど、and transfer the laboratory space to the far away from mouse space. So we never detect mouse DNA. で、2つ目が、コンパラティブですね。どうしよう。難しいな。必ず比較します。それで、OK というのを見てから、トランスファーするようにしてます。we must, we, どうやったらいいかな。後で説明します。はい。
Right, thank you. Another question? No?
Maybe I can add one thing about the device in the Prof.
Okay, please.
As Yamakawa-san has already explained, basically, ANEMONE system is following the former protocol provided by the eDNA Society. And this society has the, the regular committee where always they are discussing about the potential improvement of the protocol. And every 1 or 2 years, I think, 環境 DNA 学会のプロトコルは how many years do they take to revise it? Maybe every 1 or 2 years, I guess. Yeah. So after having this discussion with the society members, they make open the basic changes going to be made. So if you are interested in the how it is improved, we are very happy with having you in the to join this, committee and making a contribution.
I thank you very much, Professor Kondo, and thank you very much, Professor Yamakawa. And on behalf of everybody who visited Kazusa yesterday, we enjoyed the trip very much. Thank you.